Good morning, everyone, and welcome to today's webinar, How to Speed Up Cancer Genomics Multi-Omics Analysis. I'm Sharon, I'm the VP of Operations at CodeOcean, and I'm thrilled to have you here today. Before we get started, we just have a few housekeeping items. First, the session is being recorded, and we'll send you the recording after the event. Second, we'd love to get some questions from the audience today. Please feel free to submit your questions at any time throughout the webinar using the GoToWebinar questions chat, and we'll answer them towards the end of the presentation. I'm delighted to introduce our speakers for the event today. Benjamin Haibe-Kains, Senior Scientist at Princess Margaret Cancer Center and Associate Professor at the University of Toronto. Benjamin started his early career as a computer scientist over 12 years ago. Benjamin's interests include the development and application of machine learning algorithms to analyze high-throughput genomic data in cancer research. Among other activities, Benjamin investigates the use of pharmacogenomic data sets generated from large screening of drugs in cancer cell lines to build genomics predictors of drug response. An advocate of sustainable research software, Benjamin has been a power user, a customer, and a trusted advisor in CodeOcean's journey. I'm also excited to introduce you Simon Adar, founder and CEO of CodeOcean. Simon started his career in signal and image processing, hyperspectral imaging, and spectroscopy, and worked with the German Space Agency throughout his research. He devoted the last few years to empower researchers with easy-to-use computational tools so that their work is efficient, achieving faster and higher quality discoveries. I'd like you to take it from here.
Thanks, Sharon. And it's really a pleasure to host you here, Benjamin, in this webinar, in this exciting topic, a very hot topic. And what will you learn from this webinar, for the audience? We're going to talk about why and how multi-omics is useful for cancer drug discovery. We're going to talk about the challenges of data standardization and how we can solve those challenges. We're going to present essential and open tools and open data for analyzing pharmacogenomic data. And we're going to have a live demo of a compute capsule, Multi-Omics analysis, a very exciting compute capsule, a few years of research in Benjamin's lab that is now being open sourced and going to be publicly available for everyone to use after this webinar. And we're going to finish up with Q&A from the audience. So, Benjamin, we see and we hear a lot about multi-omics. People are talking about it quite a lot. What exactly is multi-omics and why is that important?
Thank you, Simon. So multi-omics is definitely a hot topic, especially in cancer research. And here we'll use the concept, the framework of precision oncology to showcase the power of multi-omics. And so the idea is the following. You have a lot of patients that have the same diagnosis. They suffer from the same cancer types. They have the same presentation of the disease according to clinical parameters. However, when you dive deep into the tumors, you realize that each tumor is really unique when you start profiling the different aspects of the tumor characteristics, such as mutation or copy number variation, et cetera, et cetera. So those are the big molecular data that we can obtain from new technologies like high throughput sequencing. And based on those data, we can really take an informed decision as to whether a given drug will be efficient for a set of patients and really kind of discriminate between responders and non-responders, which is one of the core concepts of precision oncology. And so multi-omics is this very complex data we can now generate using new technologies to better understand what makes those tumors so unique. And usually we start with genomics. That's the most established data type, omics layer, where we measure mutations, the number of copies that some genes have, potential fusions of genes, or more complex structural variations. But nowadays we also able to measure epigenomics, so all the modifications of the DNA that also influence the way cancer cells will, let's say, respond to therapy, such as methylation patterns or access of chromatin, the chromatin accessibility. And the transcriptomics is also a very well established omics layer that a lot of people use to investigate how or genes are being expressed, whether they code for protein or they're non-coding, or transcripts may be fused and again influence how cancer cells will respond to therapy. You can also do it at a proteomics level, looking at the abundance of proteins, the existence of protein complexes, and modification of those proteins. At the metabolomic level, you can basically study how cancer cells are using and generating energy that's so important for their proliferation. And above all, you also want to understand how the cells react, how those cancer cells respond to a given drug. Are their cell viability influenced by drug treatment? So multiomics is really this collection of all this very complex biological information we can now gather using a variety of recent technologies.
Great. So now we're going to pause for a three-part poll. So 3 polls. The first one, we're going to ask the audience, what are you currently using as multi-omics layers? The second poll will be about what would you look to use in the future that you find exciting as a multi-omics layer. And the third poll will be asking what are the most challenging parts of speeding multi-omics analysis. So we're going to hand it over to the poll now. This is the first question, which is what are you currently using in terms of the layers of multi-omics? And you can choose all that apply.
Okay, I'm going to give it a few more seconds. Benjamin, I don't think you'd be surprised with what we have here.
Looking forward to seeing the results then.
After we have this first poll, we're going to present the results, and then we're going to have a quick sort of a discussion, and then we're going to move to the second poll.
Okay. And here we are.
In a way, it's really expected. Genomics and transcriptomics are the most mature technologies that are also being used in clinical setting. So I think it makes sense that those are really the top 2. And it's great to see that epigenomics is really catching up. I think there is very rich information there. Proteomics and metabolomics are really key from a biological perspective, but they're also the most difficult to implement. The technology is very complex. The data are very hard to analyze. So it also makes sense that those 2 are a bit behind the other top 3.
Okay, great. Why don't we move to the second poll? Excited to see what we're going to have there. And the second poll is about what are you looking for in the future? What will be exciting for you to use in your future research in terms of the multi-omics layers? And while, sorry, go ahead, Siobhan.
We're on. The poll is running.
Okay, great. While we're getting the results from the audience, I must say, Benjamin, that for the first poll, when you and I were sort of having our prep work for this webinar, you were anticipating and predicting those results.
I was expecting the genomics to be a little bit further ahead, so it's great to see that transcriptomics is very, very close. But yes, those are really the technologies that most people are using where they're, for instance, looking for biomarkers, which is one of the topics of that presentation.
Were you surprised about the epigenomics being as high as it was?
Oh, actually, if I have to personally choose, that would be the next technology that I would use. I don't want to influence the audience here. I'm very curious to see what's going to be the results of that poll, of this poll.
Okay, let's see what comes up.
Okay.
Awesome. So yeah, that's exactly what I expected. That's also what I'm personally focusing on in terms of research. I think really epigenomics has the potential to really change the game in precision oncology. And it's very interesting to see that metagenomics is really almost as exciting as a future layer, omics layer, than epigenomics. So maybe in the Q&A session, I would love the audience to comment on how they plan to use metagenomics, which is definitely also a very important layer. But is there potential in precision oncology? I would be very interested to discuss that.
Mm-hmm. And were you expecting like proteomics to be higher than metabolomics?
Proteomics, I would have expected it so, but I think people are more used to proteomics. They understand the challenges of proteomics better. Maybe metabolomics kind of benefit from a novelty factor, and people will realize that metabolomics are probably equally or more complex than proteomics. So future will tell us.
Yeah, maybe it's the excitement about the metabolomics that influenced that number to be higher. Cool. Why don't we move to the third poll, which is what is the most challenging part of speeding up multi-omics? And we have data access, data prep, data analysis, collaboration, or other. If we didn't present anything that you might think is more challenging.
Again, if you choose order, a great topic for discussion in the Q&A session.
Yeah, and we don't want to influence the audience, but very, very excited to see what are the results there.
I'll give it a few more seconds.
Sure. And by the way, we'll be sharing the results of the polls if you want to use it in your presentations or internally. All of this will be available after the webinar, both the presentation, the recording, different artifacts, and of course the compute capsule, which is open access and open source, that you can have. You can download everything and you can use it.
Okay, here we are.
That's very interesting. So it looks like data access is still quite a bit of a challenge. Data prep is also surprisingly high, and in my experience, that's probably where I want to spend the least amount of time, but that's probably where I spend the most time. The analysis is the top one, and I guess that's a reflection of the complexity of multi-omics. Maybe if you analyze only a single layer, data analysis would be less of a problem, but I guess the combination of multiple layers makes it difficult. And collaboration is the least, but still a very high percentage. So it looks like we also capture the challenges pretty well. Only a few people choose order. So that's a very interesting kind of picture of the biggest challenges today. And we're going to discuss a few of those items during this webinar.
Yeah, and it looks like everything is around, let's say, 50–60%, and so it's across the board. There are challenges everywhere. So it shows, I think, the complexity of leveraging multi-omics technology. Okay, so let's move on now. And Benjamin, go ahead.
Okay, thank you very much, Simon. So today we're going to discuss actually a lot of those challenges and hopefully provide solutions for the audience. And we will particularly focus on this first webinar on genomics and transcriptomics, which, by the way, happens to be the most used omics layer according to the poll. And of course, the pharmacological profiles of cancer cells, because we really want to identify biomarkers to improve precision oncology. So we're focused on the three layers.
Yeah, and it looks like, you know, the multi-omics has a lot of potential and a great opportunity to benefit patients that are coming into the clinic and being given the right drug for them, that it will be efficient. And the question that comes up is, what's stopping us? What are the challenges to be able to achieve that?
That's a great question. So I think now we have the technology to generate very large volumes of multimodal data. So technology is not really the bottleneck anymore. We also have access to advanced computing, including cloud, to really analyze those data efficiently. However, as the poll suggested as well, we spend a lot of time in cleaning and processing the data so that they're finally amenable for analysis. And that's due to multiple factors, including the lack of standards, which makes combining multiple data sets difficult. And we keep reinventing the wheel by cleaning the same data sets again and again and developing the same methods to do so. And that results in a declining return on investment. So the discovery process is slowed down. You have suboptimal results because sometimes you cannot really analyze as much data as you would like. The collaboration is made inefficient. Sharing data set becomes difficult if we don't talk the same language and use the same format. So a lot of error-prone curation operations happening there. And what we are proposing here is a framework for multimodal data analysis. So first, we would like to help the community create multimodal data set in a transparent and reproducible manner using established standards. And that will definitely help us share more efficiently those data sets. So we want to make those data sets easier to find, easier to access, easier to combine, and easier to use. And at the end of the day, what we really want to tackle is the biggest challenge with it, which is how do we analyze those multi-omics data sets? Now that's also reflect what we've seen in the poll. So can we provide a computational framework that enables collaborative analysis of multiple data sets based on the fact that now we can share highly curated data sets?
So we talked about, you know, what is multi-omics? Why is it important? What are the challenges? The question is, did we see any success yet with multi-omics? Do we have proof that is actually beneficial and it's a good direction?
Yeah, so we've been discussing personalized medicine and precision oncology for a while. And before we had the ability to really deeply sequence and profile patient tumors, we were left with a relatively simple diagnosis to take our decision regarding whether a patient needs to be enrolled in a given clinical trial. So we would use traditional clinical data to do so. Not really, you know, any actionable, for instance, mutations. But as time goes on and we start to adopt sequencing technologies, then we're able to deep dive into what makes every tumor so unique, profile them at multiple levels, and then take that information to guide our decision whether a patient needs to enroll in a clinical trial X, Y, or Z. And hopefully that matching will really be beneficial for the patient as he or she is much more likely to respond. With the adoption of those technologies, we've seen that the percentage of patients enrolled in clinical trials based on those omics data, it really increases over time. And sometimes we actually find matches, but there are no active clinical trials testing the drug that we would have chosen based on the omics data. So the large investment in major cancer centers made it possible to actually assign patients to active clinical trials. But you can see that a lot of patients are still not matched. And here's a snapshot of the state of the art. Those are all clinical trials that have been performed worldwide, where basically patients were matched to the clinical trials based on the genomic aberrations observed in the tumors. And while some clinical trials were very good at matching patients to trial, you can still see that there is a lot of red. There are a lot of patients who unfortunately do not benefit from this approach.
Yeah, so basically the goal is to, you know, if I'm going to the previous slide, is to eliminate the red percentage of patients that are coming to the clinic and we cannot assign them any specific clinical trial that will be in a high probability rate will be beneficial for them to cure the cancer that they have. So we want to paint the picture here with dark blue as much as possible. And the question is, how do we do that? How do we achieve that goal? How do we match more patients to clinical trials?
So if you want to address an issue, you first need to recognize the causes of that issue. And basically, the problem is that a lot of patients come to the clinic and we use small sequencing panels. We only measure mutations or copy number variation in a small set of genes, usually a few dozens, a few hundreds. And that's sometimes we don't see any aberration that could help us guide the treatment decision. So the problem is that we also use usually a single assay. We measure the DNA or the messenger RNA or the protein. We don't really combine multi-assay that often in the clinic. And also the biomarkers, those companion diagnostics helping us predict whether a patient is going to respond to a given therapy, they are usually very simplistic. They're usually univariate. It's a single genomic aberration that we measure and link to drug response. And so the bottleneck here is really that we don't measure enough, but we also don't know enough. We don't know if a genomic aberration is actually predictive of drug response. And if you look at all the clinically approved biomarkers on the OncoKB database, you can see that only 156 genes out of the 26,000 protein coding genes have actually been associated with drug response in a clinical setting. And that's only for 100 drugs out of our current arsenal of more than 600 anti-cancer therapies. So we basically only scratch the surface. There is so much to learn. And so really the one way to address those issues is really go multi-omics. Let's measure more. Let's better understand what makes tumors so unique. And also, once you have those data, develop more sophisticated predictive models. Try to combine multiple aberrations to predict whether a patient is going to respond to therapy. Try to leverage the information to match more patients to therapies.
So I'm convinced, going multi-omics is the right way. What do I need to do now in order to generate the right multi-omics data? And I'm hearing a lot of researchers, both in academia and industry, are collecting large amounts of data, but not necessarily knowing, if this is the right data to collect. What do you think about the approach of collecting data, and what's the state of the art currently?
So that's really a very challenging and still an open question. The problem when you want to find new biomarkers, when you want to develop those predictive models for drug response, you really need to consider the relevance, but also the sample size, the number of data you can gather. And basically, if you want to help the patient, the best source of information is really the patients themselves. So you want to gather data through clinical trials. The problem with that approach is that it's extremely costly and lengthy to collect such data. So we will probably never have enough patient information for some rare cancer, for instance, to really have the sample size necessary to efficiently discover biomarkers. If you go on in the lab on the preclinical side, then you can start deriving model systems from the patient tumors and really try to challenge them with drugs and see how they react to those therapies. And if you can go with the animal models, you can go with sophisticated 3D organoids or cancer cells growing in 3D, or you can go with the venerable immortalized cancer cell lines. And as you go along that axis, you can really start to generate a lot of data. Cell lines are extremely amenable for high throughput drug screening. And so what happened is that a lot of people generate tons of data at the cell line level, challenge them with a lot of drugs and see and measure their response and use that data for biomarker discovery. So what do? We start with cell lines and organoids. Those are in vitro models, relatively simple to generate data for. Then we test the candidate biomarkers in more complex organisms, such as mouse models bearing a human tumor. And if that biomarker still works, then we go to the patient and actually test its clinical utility and validity in clinical trials. And so those data are relatively complex. Even if you use cell lines that are relatively simple cancer models, you can basically start from a large collection of cancer models, a large collection of cell lines. You can profile them at the molecular level to really try to best understand their uniqueness, what are the features that makes each of those cancer cells unique, and you do so at the multi-omics level. That's kind of our input feature X, matrix X. Then we profile those cell lines with drugs and basically see whether the viability of those cell lines are impacted as you increase the drug concentration. If you have a cell line that's very resistant to a drug, you have this flat line, horizontal line at 100%. And if the cell lines are responding to the therapy, you can see that the viability is decreasing as you increase drug concentration. That's our phenotype, that's the Y. And the biomarker discovery process is really combining those two complex data sets to find a function that, based on the omics data, would help you predict whether a certain tumor, a certain cancer would respond to therapy. So that function f of x is really what makes a biomarker. It could be very simple, it could be a single feature or linear combination of features, or it could be as complex as a deep neural network, for instance. And the beauty in the field of preclinical cancer pharmacogenomics is that we got access to tons of data. And it all really started in the end of the 90s with the National Cancer Institute Developmental Therapeutics Program, where they screened a small set of 60 cell lines with a lot of drugs. And you can see that both industry and academic labs kept generating data using more cell lines and screening more drugs and release them to the public domain. So you can see that GlaxoSmithKline, Pfizer, Genentech, AstraZeneca, and Novartis all contributed public data sets for the research community to leverage, as well as their competitors as well. You can see that academic labs have done the same along the years. So it's really a massive amount of data that you can now be using, you can now use for biomarker discovery.
You know, I remember from my PhD that I was collecting different types of data sets, open data sets from various sources, and it was a great challenge. Everyone was measuring differently, the formats were different, the metadata was different, and I spent a lot of time basically cleaning and organizing in a way that I can combine various data sets into one coherent analysis. You know, I'm looking at this, there's a lot of data here being generated in all sorts of places, a lot of potential here. I wonder, you know, how easy it is to actually combine those data sets.
So your experience during your PhD matches very closely what we see in cancer research as well. All those data sets are very unique and very different in many ways. They don't use always the same standards. The way the format they use to release the data is very often different. They sometimes lack metadata about the experimental design or how you treat the phenotype. And so at the end of the day, all those pharmacogenomics data, even so it's a massive compendium, they're very difficult to clean and create and align, and they are not directly amenable to statistical and machine learning analysis. So really what we need to do now is to improve the fairness of those pharmacogenomics data. How do we make them more easier to find, access, more interpretable because we use standards, more reusable? And maybe something also very important that's often overlooked is how do we make this whole process more reproducible so that people can easily scrutinize, improve, and reuse in the future. And so that is really what my lab has been focusing on for many years. There are many brilliant trainees that have worked on that problem to really try to do it in a way that the community can reuse. It doesn't have to go through the same process, same pain again. And so we developed the Pharmacogx Toolbox. It's an open source R bioconductor package that really help the analysis, the integration of all those data so that you can download data and directly focus on the analytical part. And so in the lab, we've curated 8 data sets so far, and there is much more coming. And that represents a very large panel of more than 1,600 cell lines coming from 41 different tissue types. It's almost like almost 1,000 compounds that have been tested on those cell lines. and that represents more than 1 million drug dose response curves. So the combination of those data sets really represent a massive data compendium for biomarker discovery. And so we are going to discuss today one of those data sets, one of the largest one. It's called the Cancer Cell Line Encyclopedia. So it's a collection of over 1,000 cell lines from their broad cell line factory. And they basically team up with Novartis, who did a lot of drug screening on these very large panel of cell lines. And the Broad Institute has also provided those cell lines with a very, very diverse set of omics technologies: mutation, copy number variation, gene expression, methylation, proteomics, metabolomics, as well as chromatin accessibility. So it's a really complete data set. And we've created that data so that the integrated object can be analyzed using our Pharmacogx package.
Yeah, so before we dive into the most exciting part of the webinar, which is the actual compute capsule, which is open, just a few words about what is a compute capsule, what is CodeOcean. So CodeOcean has developed a technology we call the compute capsule, which is basically a secure and standard package that comprises three the most essential elements of any computational work. And it's the code, the data, the computing environment in form of Docker, and the associated result. And a lot of people are asking, so what's the difference between this and Docker? Docker is only one component of the compute capsule. And the technology is built in a way that you can attach data sets. It could be as large as you want. It could be terabytes of data as well to one computational framework. There's a repository of compute capsules. We have a public open cloud for anyone to basically log in and use. We have mostly academics using it as part of the publishing in journals like Nature and American Association of Cancer Research and others. And everyone can take a capsule that is published, pull the capsule, reproduce it, change each and every one of the elements of the compute capsule. Could be code, data, and packages, new packages if you want to. Now you have a new compute capsule, you can put it back to the repository for the community to be used. There's also a private use, so you can install CodeOcean in a private cloud if you want to. But everything that we're going to show you now is on our open public cloud, and Benjamin worked on this capsule and published it, I think, yesterday. The product itself in CodeOcean has two main components: a computational workbench that you can use to create, organize, and share your work with all the different open source tools that you like to use, like Jupyter, Python, R, RStudio, any programming language that you want to. And then the public repository is built in FAIR principles. We actually call it FAIR squared principles, which is findable, accessible, interoperable, and in addition to reusable, it's reproducible. So I'm just going to hand it over to Benjamin now for the most important and interesting part of the webinar.
Perfect. So thank you very much, Simon. So as Simon said, the code and the data is only part of the story. What we really need to put together is a platform that makes it very easy to analyze data and generate results. So do you see my window?
I think we see a blank window.
Oh, okay. Sorry for that.
Now we see your whole screen.
Okay. Can you take over and give me the permission to share my screen again?
Yeah, maybe you could just magnify the window, the Firefox window.
Does it work now?
Yeah, works.
Okay, perfect. Sorry for that. So as Simon said, we really need to put all those pieces together to make it very easy for the community to be able to reuse all those artifacts, the data and the code. So we basically use curation here to basically load the data and generate some code and be able to share that with the community. So I would like to present you what the interface looks like. On the left side, you have all your different files. Most importantly, you basically have a very easy way to attach data to your capsule. So for instance, here, I selected the cancer cell line encyclopedia data asset to load into my capsule. And on the right side, you can basically change the code, clone the capsule, commit, and use a different series of tools here that are very common in the bioinformatics community. And the capsule itself, it's a markdown, so it's a combination of text and code, as you can see here, that will basically do a lot of the, basically all the loading the data, loading the software, and do the analysis for you. And that is has been packaged into a Shiny R Markdown to really make it very easy to probe the data set. So I'll present you the results of that capsule. So first of all, the disclaimer about access to the data, I think credit needs to be given to the data generators, and the Broad Institute has done a fantastic job at sharing all data and metadata with the community. So here you have a few explanations about what pharmacogenomics is, how you can use the Pharmacogx package that we have developed in the lab to analyze those data, and how you can access the cancer cell line encyclopedia data set. So you basically load the library, read the data asset that you've loaded in curation, and if you print that object, you already see a series of information that's quite interesting. What kind of data is part of the data set, how many cell lines, how many drug dose response curves, for instance. But that's a very complex data set, and that's kind of what makes multi-omics data sets so unique. And so if you want to start probing those data objects, because they've been created and standardized, you can actually easily do so. So if you go on the cell lines and you want to know, well, what's the sex distribution of the cell lines, or what's the tissue distribution of the cell lines, you can easily do it here through interactive menus. You can see we have a lot of lung cell lines and lymphoid and hematopoietic cell lines, for instance. You can actually gather more information about those cell lines if you wish so. For instance, if you want all the bone cell lines and you're interested in specific sources, let's say all the bone cell lines from ATCC, it's only a subset of that table, but all the information is there, including histological type, sex, and source, for instance. And that's only for the large panel of cell lines. What about the drugs themselves? As I said, Novartis generated a lot of drug screening information for many drugs, and you can see here that those drugs, not surprisingly, are majority from mostly from Novartis itself. They were interested in their own drug, makes sense. But they also tested all drugs from competitors to basically provide references and potentially improve the biomarker discovery process as well. Those drugs have also a lot of metadata annotations directly accessible from the data object. So for instance, if you're interested in the kinase inhibitors or the cytotoxic agents, you can see the company who produced the drug, the target, the annotated target and mechanism of action, the class, whether it has been already launched in clinical trials and what's the highest phase, and whether it has been approved by the FDA, for instance. So now we can easily probe cell line, learn about the cell lines and the drugs that are in this multi-omics pharmacogenomics data set. But what about the multi-omics themselves? So those data are also obviously very complex and very high dimensionality. A lot of genes, a lot of measurements have been generated here. And so to first present the DNA information, so the mutation as well as the copy number variation, either deep deletion or amplification, we can use what we call an oncoprint, what the community is calling an oncoprint plot. So it's a plot where you have all the cell lines in columns, you have all the genes in rows, and you have the mutations represented as this little green dot. Then you have the blue and the red cells representing the copy number of that gene. So whether the genes has disappeared from the genome of the tumor with a deep deletion, or the gene has been amplified. There are multiple copies of that genes. And you can see here that if you, for instance, select the lung cancer cell lines, but you can select any others, and you choose a different a given pathway, just a set of genes of interest, you can see here that you have a lot of cell lines with deletions, a few cell lines with amplification, but there is no real patterns of co-occurrences. So if you have an amplification of the gene, you often don't have a mutation in the same gene. So that basically depicts the complexity of the DNA information in those cell lines. Now, if you want to look at the genes, yep.
One thing that we did not mention is everything that you're showing here could be used, there are different personas here that can use this: bench scientists, bioinformaticians, computational biologists. So everything that you're showing here, it's a web application that you don't need really any knowledge of coding. It's ready to be used for non-coders as well. And if you're a computational biologist, all of this is open source. So you can, if you want to change what you're saying here, you have the power to do so.
Correct. So I think that's really, for me, the beauty of those capsules is that it's basically useful for different groups of people. If you want to dive into the code, you can easily clone it and modify and have different menus and different ways to visualize the data. If you just want to have a quick look at what this data contain, then you can look for your favorite cell lines or favorite drugs and see whether you have data of interest there. And then you can collaborate with people who are very good at programming, for instance, to actually add functionalities. And this capsule is a living document in the sense that it will be improved and enriched over time, as you'll see at the end. So we already saw how we can visualize DNA. Here's a way to visualize gene expression with a more traditional heat map, where you have, again, all the genes in rows, and all the cell lines in columns. Blue representing a lowly expressed genes and red representing a highly expressed gene. So you can see that some genes are already lowly expressed across the board or highly expressed across the board. But there are some genes where you see a lot of variation, and that's really where there is potential for a biomarker to see whether that variation is actually associated with drug response. And talking about drug response, those are also rather complex measurements where basically the experimentalists measure the viability of the cell and kept increasing the concentration of the drug given in a given range. And it started at 100, so the cells are growing as fast as the control. But as you start treating the cell lines with the drug, you can see that sometimes there is a dramatic drop in cell viability. And here's an example of ERBB2 positive breast cancer cell lines treated with lapatinib. And lapatinib is actually a targeted drug whose target is ERBB2. So it makes sense that cell lines learn to rely on ERBB2, and therefore when you inhibit it, the viability really drops. But some other breast cancer cell lines, such as MDMB157, is actually rather unaffected to the treatment with that drug. So it really gives you very different phenotypes here. We have 1 million of those drug dose response curves in Pharmacogx. In this dataset alone, there was more than 10,000. So you can actually choose your favorite cell lines and your favorite drug and display that drug dose response curves to actually see whether there is a phenotype of interest. But those are just a series of points here. What you really want to do is to model that curve to kind of be able to summarize its behavior. And if you do so, we implemented this well-established log logistic model. You can see that we can basically take the viability measurements, they are represented here as the dots, and basically fit a sigmoid model where we can basically estimate the slope and estimate the concentration you need to kill 50% of the observed inhibitions. You can even compute the area above or under that curve as a way to kind of summarize that curve into a single number. And that's very important for biomarker discovery. Most machine learning methods are actually very good at predicting one value, whether it's a discrete value or a continuous value. And so we need to be able to estimate, to model all those curves. So here, for instance, using a simple function, compute AUC or compute IC50, you can actually compute those values directly from the curve, and you can even display them on the curve itself. So here, in that case, you can see that the AUC is 0.5, which basically means that a lot of the viability has decreased as you increase the concentration of the drug. So that's an example of a very sensitive cell lines to lapatinib. That's a lot of data too. For each cell line and drug pairs, potentially you will have a series of statistics, whether it's the area above the curve or the IC50 or the hill slope. You can basically display all of them in the cell lines of interest, and using a complex heat maps where you have the drugs in columns, you have the cell lines, the drugs in rows, cell lines in columns, and you can basically display additional information such as the class of the drug, the company who produces the drugs. You can, for the cell lines, for instance, you can display the tissue type or the sex, and basically look for any pattern here. So you can see, for instance, like all the bladder urinary tract cell lines are very sensitive to panobinostat, and some cell lines are very resistant to basically to a lot of therapy. So you can really start understanding the variation of the data and use that for biomarker discovery. And so at the end of the day, what we really want to do is to jointly analyze those data to find potentially new biomarkers. And in this first version of the capsule, we've implemented something very simple. It's just a linear regression model, taking a series of features as input, correct for tissue type when you look at multiple tissue types, and look whether a given aberration is highly associated with drug response. And you can see here, you can choose the drug, you can choose whether you want all the cell lines or specific tissue types, some pathways of interest, just to make it super quick to compute, and then whether you want to focus on mutation, copy number variation, or gene expression, and which type of drug response summary you want to use. So in that case, we use the area above the curve that we recomputed within Pharmacogx. And you can see that if you do this analysis as expected, we see that EGFR mutation is actually very significantly associated with response to lapatinib, to erlotinib. And that's fully expected. It's a very non-biomarkers. It happens to be the target of the drugs as well. So that's kind of confirmed it. If you want to look for other data types, you can do a similar analysis and get the results in an interactive way. So the copy number variation, actually, the EGFR is also associated. So now you start to understand that it's not just a mutation, it's also the copy number variation that's associated. If you look at the RNA level, however, that might not be the case. So the top hit might not be EGFR, even so it is in this case. So EGFR seems to be a very good biomarker that seems to be associated at all omics level, which is very interesting. If you want to do de novo biomarker discovery, sorry,
we're starting to get a lot of questions, and we do want to give some time for questions from the audience. So maybe just a few last words on the open capsule, and then we move to Q&A.
Perfect. So I told you before we focus on pathways, you can actually do a de novo analysis as well. You can look at all the genes that have been measured in that data set and find associations such as, new, for instance, resistant markers of lapatinib at the RNA level. So that basically showcases how you can analyze all those omics layers separately. What's coming up in the next version of the capsule is really how do we combine those multi-omics data to discover something completely new that might be hidden if you analyze every layer separately. So we'll showcase the example of how you classify drugs, classify cell lines, and how you build multi-omics biomarkers in an interactive manner. So with that, that's the end of the demo. This capsule is publicly available. You're free to duplicate, download, use it in your own infrastructure, use it in CodeOcean. It's completely open. If you have suggestions on how to improve it, they're obviously all very welcome as well.
Yeah, and by the way, if someone from the audience needs some help from the CodeOcean team or from Benjamin, you're free to reach out to both us and Benjamin on that, to help you get started. One question that came from the audience, is for you is help them understand what is the Pharmacogx and which part is in CodeOcean. So make that a little bit clearer.
Sure. So Pharmacogx it's our bioconductor package. So it's open source, you can download, you can use it in your R session. You don't really need codation in any way. This is really part of the R bioconductor ecosystem. However, codation makes it very easy to basically build a virtual machine, install Pharmacogx, and use it with data sets. So basically, Pharmacogx is a package you can use on your own, or you can use on a codation platform. When Pharmacogx is used to analyze the pharmacogenomics data, as you saw in the capsule, it's rather simple to access all that information.
Yeah, and just to add, you can export the capsule, so you will have all the relevant components. You can download the code, you can download the Docker image, a Docker file. You can recreate the environment if you want to. You don't have to use CodeOcean. CodeOcean basically helps you to get a package that is easy to reproduce even outside of CodeOcean, and also the data. So
Simon, I'm going to jump in here with another very interesting question that we got from the audience, and we are kind of transitioning into the Q&A. That's a good opportunity to remind everyone we're still collecting questions. One question we got from Evan was, what is your thought on the match miner developed by Dana-Farber? I think that's a question for Benjamin.
Yeah, so match miner is a fantastic framework to, as I said, match patients to clinical trials. And it's basically based on approved biomarkers. So you can encode your eligibility criteria for the trials. You can input the sequencing data or other omics data that you've generated through the hospital. And based on those two pieces of information, it will basically tell you, based on those approved biomarkers, this patient is eligible to clinical trial XYZ, and that's heavily used, I suppose, at Dana-Farber, but maybe other institutes as well, to kind of help the clinicians guide their decision on which trial the patient should be enrolled in. What I would love to do what I would love MatchMiner to do is to be able to expand the portfolio of biomarkers so that it can match even more patients. So there is an interesting interaction, a collaboration between the research and the clinical side, where we need to expand the portfolio of biomarkers so that we can match more and more patients. And MatchMiner is actually a software framework that really makes it easy for clinicians to use this knowledge about biomarkers and assign patients. So again, a very important piece of software. Actually, in my own research, we are working into seeing how we can use an expanded portfolio of biomarkers and use MatchMiner to help clinicians make better decisions.
Another question that we have here is regarding multi-omics. You know, we're looking at pharmacogenomics and pharmacology. Besides that, what are the other use cases of multi-omics? So,
if you have the resources and there is the need for it, multi-omics is a beautiful tool to investigate many other questions. It could be used in agriculture if you want to optimize, the way a crop is growing. You may want to use it in mining to study the effect of mining activities on the environment. So it's just a tool to probe the biology at a very, very deep level. And it's basically used in many other areas than precision oncology.
And another question regarding the future beyond multi-omics, what are the new technologies that you're seeing as exciting? I heard about taking multi-omics to the spatial level.
Yeah, so even so, it's kind of multi-omics is going to be applied more and more often, and we're already thinking about the next step. That's human nature, I guess. Always looking forward, and I think spatial omics profiling, such as spatial transcriptomics, really trying to understand not only what makes a tumor unique, but where those cells expressing those genes are in the tumor could really be a game changer. Single cell omics profiling as well, understanding that a tumor is not composed of all identical cells, but there is a lot of heterogeneity across the cells. So single cell omics and spatial multi-omics are really the future, even so those technology needs to mature a lot for them to be applied on a large scale. But that's really, for me, those are two very exciting technologies, as well as liquid biopsies, not only doing it from tumor material, but also potentially blood, to really better understand and monitor the response of tumors to the treatment.
Yeah. Another question is, the multidimensionality nature of the data reminds me a lot of tools that you can use, like mathematical hammers for dimensionality reduction, diffusion maps, all sorts of machine learning tools that people are using, not necessarily in the bio space. Are there any? Do you see a lot of collaboration there to tackle multi-dimensionality of the data?
And that's a great question. So I think there are two schools, probably many others. One is really trying to model the biological mechanisms that we know, and basically look for interactions between the different layers. And that basically requires very deep understanding of the dynamics of the system. But that's where probably mathematicians are really shining in the field of systems biology. And another school of thought is really to try to get enough data to learn that from scratch. And that's where machine learning techniques that extract signals from multiple layers and potentially look for complementarity or convergence of those signals are really, really efficient. So I suspect it will converge as we know more and more about the biology and the dynamics of the system. This knowledge will be included in more sophisticated machine learning techniques. But for the moment, those are really kind of the two approaches that people are using.
Okay, that's great. I just want to mention we're getting a lot of questions on how to use the capsule. I think there's a lot of excitement of everything that you've shown. So we're really happy to help everyone. There's a chat item on the CodeOcean application. So you're more than welcome to approach us, and we're going to get you up and running with everything that you need.
Simon, maybe before we're wrapping up, I do want to jump in with another question that we just got from Christina. What about other disease? How can the capsule that Benjamin just presented help there?
Yeah, that's a great question. It's really the hope, and the hope is to make that framework as reusable and as flexible as possible. So for the moment, we focus on cancer pharmacogenomics because that's what my lab has worked on for many years. However, it could really be used with other multi-omics data that have been generated with many other diseases. And as I said, even fishing and agriculture and mining in many ways. The concept of looking for associations between multi-omics data and a phenotype, whether it's drug response or something else, that should be reusable. So if you're interested in if you have access to a very interesting data that's for Alzheimer or Parkinson or a completely different disease, we would be happy to kind of work with you and see how much of this capsule you can repurpose for your own research question. And I truly believe that that's the power of those open source capsules, to really let people leverage and modify the way they want. From a machine learning perspective, there's a lot of commonalities that could be leveraged.
Yeah, for sure. So we have 2 minutes left. I just want to wrap things up. And do you see my screen? We have a lot of resources that we collected here, and we'll share that after the webinar, both from BHK's lab and publicly available. But we do want to wrap things up. Maybe Sharon, maybe you can take that one.
Sure. So I think what we're seeing on the screen right now is Benjamin's email. I'm going to share Simon's email as well in the chat. I'd like to thank everybody for coming in today. It was great having you. We'll be in touch in the next few days with the recording and a link to the capsule. On behalf of CodeOcean and our presenters, thank you again. Have a wonderful day.
Thanks, everyone, and thanks, Benjamin. Really, really exciting sort of open source package here.
Thank you very much, Simon. And all feedback is welcome. Let me know if you want to see a new functionality. I'll do my best to implement it or work with smart people to implement it. So we'll make the capsules better over time with your feedback.
Yeah, that's actually a great point, because this is a work in progress. I don't know if the audience noticed that we are mentioning that some of the things are coming soon. So this is something that Benjamin, your lab, and you're working on, and we're happy to facilitate. And if there's some questions from the audience that they would like to see a specific direction, actually that's something I think that you find very, very exciting to get as feedback.
And all credits also come to the many trainees who worked on that along the years. So we'd like to thank all my lab members as well.
Absolutely. All right. Thanks, everyone.